Alright, so this audio lecture is going to cover the development of B lymphocytes, or B cells. This is covered in chapter 6 in your textbook. When we look at the development of B cells, it can be divided into 6 different phases. The first 3 phases are going to be happening in the bone marrow. So early stages of development, again, are in the bone marrow for the B cells. The last 3 phases, phases 4 through 6, this is when the B cell has moved into secondary lymphatic tissues such as spleen, lymph nodes, pilar patches, and that's where it goes through additional developmental stages before it can become a mature B cell where it's going to actually secrete antibodies. Okay, as I go through this, I just want to note in phase 3, the textbook calls this positive selection, or has it labeled as positive selection. This is a pet peeve of mine because there is a term, a process called positive selection that we use with T cells, and this ends up causing a lot of confusions for students. So their use of positive selection is the counter of negative selection here. So it is not the term positive selection. So if you see on the exam or something a question about positive selection, it is not referring to B cells because B cells do not undergo positive selection. And again, that's why it really irks me that textbook authors put this term positive selection is, because if you go into your glossary and look at positive selection, it's going to talk about T cells and how they are selected for their T cell receptor being able to interact with MHCs. You'll remember from our last unit when we talked about the B cell receptor, it does not have to interact with an MHC, and so we do not need to undergo positive selection for B cells. Okay, and again, that's a whole long-winded way to say, why don't they just say the lack of negative selection? So the basic stages of development is we're going to generate the B cell receptor, right? And so this is where we are selecting for the heavy chain and the light chain. So this is that repertoire assembly, meaning assembling of the B cell receptor. That B cell receptor has to then go through selection. It goes through negative selection and only negative selection. So it goes through negative selection, and what negative selection is where the B cell receptor is going to be basically tricked into responding to self. And if it does that, it is deleted. So what you want is a B cell receptor that does not recognize self. So you want a negative, right? So hence why it's called negative selection. So for negative selection, this is again where the receptor, and in this case for the B cell, is presented self. If it recognizes self, it gets deleted. So it doesn't move forward to phase 3. The cells who pass negative selection that do not recognize self are expanded, okay? And this is what phase 3 is an expansion of that population. They are promoted, they get different expression of different molecules on its surface, so they are now this immature B cell, and they can leave the bone marrow and go to secondary lymphatic tissues. Many are going to go to the spleen as their next stop, and for phase 4. So in phase 4, they're actually going to go through another round of negative selection, which your textbook doesn't really emphasize. And in that second round of negative selection, again, what we're just doing is doing another check. This B cell has the potential to go out and interact in secondary lymphatic organs, also in the blood, with antigen, and we want to make sure that antigen isn't self. So initially when it goes to the secondary lymphatic organ, primarily spleen, it's going to go through another round of negative selection. If it passes that, then it would be considered a mature B cell, and it can then go and interact with material, and if it becomes activated, so in phase 5, this is where it becomes activated, it'll undergo clonal expansion. And we saw in our last journal club, clonal expansion really just is proliferation, right? So an increased number cloning, so it's going to have the same receptor that it's gone through these 4 phases with, is going to be passed on to these clones, okay? And that B cell receptor is, because it's become activated, saw its antigen. So in phase 6, it can now secrete antibodies, so it's undergone class switching. It's undergone somatic hypermutation potentially, and then it can again interact with antigen and then play the role of the B cell, producing antibodies, producing cytokines. It can, other immune cells could sense through their receptors. Okay. So just to give you an idea, a vague idea of location here. So we got, these B cells going through development in the bone marrow, which you see the bone indicated by the yellow. And then it's going to move, go into circulation. So the arrow going to the heart is supposed to indicate going into circulation. And then go on to secondary lymphatic tissues. Many other textbooks will mention it going to the spleen for a second round of negative selection, and then onto other secondary lymphatic tissues, such as lymph nodes, pyeloid patches, and such. So just be aware that if you're using other textbooks, and also when I talk about it, when I mention the second round of negative selection, you should know that this is happening in the spleen. So we can, we give staging to B cells depending upon where they are in their development. So keep in mind that B cells are going to originate from the pluripotent hematopoietic stem cells. So remember that back from Unit 1. So HSC gives rise to all leukocytes and lymphocytes, and so B cells are no exception. So we're going to get from the hematopoietic stem cell, we're going to get the common lymphoid progenitor cell, and you can see how the surface markers on those have changed. So we now have expression of CD10, and then we're going to get the B cell precursor, and so again expression changes. And then we get the pro-B cell, and we'll look at what's happening at these different stages. But the emphasis on this figure is the changing of surface markers. And what they're showing is this like increase of these markers, right? So we're adding a marker with each stage. Keep in mind that some markers are going to go away. So there's going to be certain markers that code for stem cell likeness. And so those markers actually get downregulated as the B cell goes through its development and moves towards a pro-B cell stage. Okay, so just be aware of it. It's not always an addition of surface markers. Sometimes it's a deletion. So when we're doing experiments, if we want to look at what stage of development the B cell is in, we can use these markers using antibodies. We can do FACS analysis. So cell sorting, where we can sort the cells. We can also do genetic analysis too. We can, look at RNA production, protein expression and such too. We can also use knockout and knock-in mice to determine the roles these different molecules have. Okay, but just keep in mind that these surface markers aren't there just for our identification purposes. They also serve the purpose of acting as receptors. So when their respected ligands bind, the respected molecules bind, that is going to lead to activation of cell signaling pathways that lead to differentiation of that hematopoietic stem cell to a common lymphoid progenitor cell, to the B cell precursor, to the pro-B cell. So when we look at the different stages, we really want to think about the B cell receptor, right? So think about last unit when we talked about somatic or gene segment recombination. We have these different stages, and we can look at the heavy chain, we can look at the light chain, where it is in an arrangement, and then the overall status of the B cell receptor, the Ig. Okay, and so you can see here the stem cell for both those genes for heavy and light. It's just going to be what is germline encoded, so what it received, what the majority of the cells in the body have. And then at the early pro-B cell stage, that's when we get DJ rearrangement, right? So the diversity and the joining segment coming together. So RAGs binding on there, cutting, RAG2s cutting, or joining those together. And then in the late pro-B cell stage, that's when we have the variable and the diversity being having the recombination events between those segments, okay? So then at the large pre-B cell stage, that's when we have, for our heavy chain, we have that VDJ exon created, okay? So we would synthesize that heavy chain. In the small pre B cell stage, that's when we move to the light chain and it undergoes the VJ rearrangement. So again, this is where RAG1 is going to bind on, RAG2 is going to do the cutting. So we then have a variable and a joining segment that are combined together. Then at the immature B cell stage, we now have both the heavy chain and the light chain expressed. And so those would be synthesized and we would have IgMs on the surface. So we'd have functional B cell receptors that still need to go through the negative selection process and also then go out into circulation. So again, really think about, last unit and then linking what are the events that we talked about last unit that are happening? Okay, what's happening at these different stages? So again, keep in mind that the heavy chain is going to be synthesized first. So you have to go through the 2 rounds of arrangement. So D and J and then V and D. And so you can synthesize your heavy chain. So during the pre-B cell stage, that's when you're going to have your heavy chain. And then you start working on the light chain so that by the time you're an immature B cell, you have both your chains selected. So thinking about some of the surface markers on the B cell during these developments, and again, kind of keeping in your mind, like what's happening to the B cell receptor. Because remember, the cues to undergo those processes of development of the heavy chain, development of the light chain, are due to cell signaling events. And those cell signaling events come from the receptors that are on the surface of the B cell, the developing B cell, and components that are given to them or produced by stromal cells. So stromal cells are support cells, so they're going to produce different factors which the B cell needs in order to receive the cues in order to undergo the development, right? So it's kind of a chicken and egg kind of thing. So one of the interactions that you should know about is c-KIT, or referred to in your textbook as just KIT. This is a receptor that binds stem cell factor, or SCF. Okay, so SCF is stem cell factor, and it's going to bind to c-KIT. And so early in the development, that pro-B cell is going to be expressing c-KIT, but then over time you can see how there's loss by the pre-B cell stage of c-KIT expression. So it's not going to be able to sense that stem cell factor. Okay, so that initial, receiving that signal, receiving the binding between the receptor and the ligand, keeps the B cell into kind of a stem-like state. But as it's developing its B cell receptor, it's kind of been committed to that lineage, right? So it can lose that receptor. Okay, and again, it's the stromal cells that are supporting that B cell by producing different factors. You can see even early on, they're going to be producing adhesion molecules. So we have CAMs there that are going to allow for adhesion to that bone marrow stromal cells, so that they're going to be guaranteed to get the factors that are necessary. Over time, as the B cell develops, so that when it's in that late pro-B cell stage, pre-B cell stage, it's going to start expressing IL-7 receptor. That's important because IL-7 gets produced, and that's a big cue for it to differentiate and become that immature B cell. Again, keep in mind that at the pre-B cell stage, that's when it would be expressing its heavy chain. It's actually expressing a surrogate light chain at that point. So again, if we look at the pro-B cell stage, we can divide that into an early and a late stage, and this is where the heavy chain is undergoing its rearrangement. So during the early stage, that's when you have the DJ segment. If that arrangement is productive, then it moves on to doing the VD rearrangement. If that's productive, then we can move on to a pro-B cell stage. If it isn't, then they have to go back to kind of the drawing board and has to redo the VD rearrangement. And if that still doesn't work, if that doesn't fix the heavy chain, so you have a functional heavy chain, then that's going to signal the cell to go into apoptosis, right? You kind of already cut out, you've done 2-3 cuts, you've altered the DNA such that there's really no coming back from that. So again, apoptosis pathways would be activated, and about 50% of cells go into apoptosis. So 50% of those cells that never become B cells have done something wrong. And not necessarily wrong, but remember, this is random, right? Arrangement is random, and so randomly, by putting 2 DJ segments together and then a V segment onto that, it just doesn't code for something that's useful, right? That's going to make a functional heavy chain, and so the cell goes into apoptosis. Now, in some cases, this second round of VDJ segment rearrangement can lead to a functional heavy chain. And at that point, again, that cell would survive and be able to go into the pre-B cell stage. During the pre-B cell stage, this is where a pre-B cell receptor is assembled. And so you can see here how we have this surrogate light chain, right? And so this is not a functional light chain as we'd recognize it. This is a kind of, let's test this out. And again, really what we're testing at the pre-B cell stage is that heavy chain. Is this heavy chain functional? Do we have a transmembrane portion to it? Can it interact with these co-receptors? And does it have the potential to interact with a light chain, right? So again, the pre-B cell stage is really focused on the heavy chain and making sure the heavy chain has potential to be functional. Then we're going to go back and make a light chain, right? So surrogate light chain is just a surrogate, it's just a fill-in to assess the heavy chain. So then with the light chain, we're going to undergo gene segment recombination or somatic recombination. So we are going to select for a V and a J randomly to come together to make that coding exon, the coding for it. If that is non-functional, the heading, the variable segments that are before the variable segment that was selected are still there. So the B cell has the ability to go back and say, nope, the variable segment we originally picked didn't really work. Let's try a different pairing. So you can see here the first initial of taking variable 1 and then the second joining didn't give a functional light chain. So it's going to go back and it's going to use variable 2, and it's the third joining that was left. And okay, well, that still doesn't work. So let's take variable 3 and the last joining that we have left and make another one. And that one finally gives us a productive light chain. So again, they're able to kind of go back and edit the light chain if it's non-productive several rounds. And keep in mind, first they're going to use kappa. And if kappa doesn't give a productive light chain, then they can move on to lambda. Because at this point, after they've gone through these multiple rounds of rearrangement that have been non-productive, again, it's too far gone, but you have a good heavy chain, right? So if you get to this stage where we're picking the light chain, you have a good heavy chain. And you don't want to go, just say, all right, cell goes into apoptosis. We want to try to fix it, right? So it's going to, under kappa, goes through several rounds of trying to fix that non-productive light chain. And if that doesn't work, that's why we have a second gene, right? That's why we have lambda that we can go to. So again, when we're in that pre-B cell stage, again, initially we're checking that heavy chain, then we're moving on to the light chain. And so you can see how if we have our first arrangement for the kappa gene and that it gives us a functional receptor, then we're good. We're fast-tracked to immature B cell stage. But if it isn't, then we're going to go through additional rounds of the kappa. If it still fails, then we'll move on to lambda, and if lambda fails, then we'll go into apoptosis, okay? So you can see here how we could end up with a B cell with a receptor that has the heavy chain, which is going to be referred to as mu, and then kappa for the light chain. Or we can end up with a cell that has mu, the heavy chain, and then lambda for the light chain, right? So kappa is going to be expressed first. It gets to go through some editing if needed. If that doesn't work out, then we go to lambda. Again, we can edit lambda. If that doesn't work out, then it doesn't matter. We vested too much energy. We have a good heavy chain, but we've already vested too much energy in that cell trying to get it to work. It's just not going to work. So though there's many characteristics of the heavy chain that makes it a good heavy chain, there's characteristics of it of not being able to bind with the light chains that doesn't end up coding for a functional B cell receptor. So in the bone marrow, there's 2 checkpoints to determine if the B cell can continue with development. So again, the first one is during that B cell receptor stage, and that's where we're checking that heavy chain to make sure the heavy chain is functional. If that heavy chain cannot make a pre-B cell receptor, it's going to go into apoptosis. The second round is where we have our light chain assembled with the heavy chain, and so we're expressing our B cell receptor, or at least our potential B cell receptor. If that receptor doesn't work out, so if that pairing of the light chain and heavy chain doesn't work out, then the cell will go into apoptosis. If the pairing does work out, then the cell moves on to the immature B cell stage, and that's where it can undergo negative selection in the bone marrow prior to going to secondary lymphatic tissues. So keep in mind, in these stages, there's a lot of change of expression of different proteins. So you can see on the left-hand side how there's different proteins labeled, and then what their function is. And then in the main part of this figure, you can see how there's the red area shows increased expression of said proteins. And if you think to what is going on at these different stages and what these proteins do, it makes sense, right? So we're going to pick on RAG1 and RAG2 because those are enzymes you're familiar with and what their function is. So you know that they're important for gene segment recombination. And we know we have multiple gene segment recombination events happening during the B cell development. So we're going to have recombination events happening when the heavy chain is being made, right? So where is that happening? That's in the pro-B cell stages, right? So you can see how that red indicating increased expression of RAG1 and RAG2 coincides with that pre-B cell stage when we're going to end up having these events occurring, gene segment recombination. This other time we have gene segment recombination is with the light chain. So you can see the expression there coincides with when we have the light chain undergoing recombination events, right? If we look at things like lambda 5 and V pre-B proteins, don't expect you to memorize these ones, but you can see that they're involved with the surrogate light chain, and so you can see how that coincides with when the surrogate light chain is being expressed, and that's in the pro-B cell stage into the pre-B cell stage, right? So again, if you think about these different markers and what their function is, it makes sense where we're seeing these changes in expression levels. So once the cell has reached immature B cell stage, it's going to undergo negative selection. So again, this is where it is going to be presented with self-antigen. So you can see the stromal cell here is expressing self-antigen, and then the B cell is going to engage with that if the receptor recognizes it, and if the receptor recognizes it, that B cell isn't going to be allowed to have the signals in order to leave the bone marrow, and it'll end up going into apoptosis. So you can see on the left-hand side how this B cell does not engage with these self-antigens because its receptor does not recognize those. So that immature B cell is able to move into the blood, and it's going to express both, it's going to co-express IgMs and IgDs. So that B cell that did recognize self-antigen, it does have, the opportunity to fix itself, because again, we've invested a lot of energy into it to this point. And so it can continue to rearrange its light chain gene. So if it passed to this point with kappa, it can go back to its kappa genes and again pick new combinations to see whether that light chain pairing would be sufficient so it would no longer recognize self. If it had passed to this point with lambda, then it's going to go back to its lambda gene and see whether it can come up with a combination for the B cell receptor that would make it so it didn't recognize self. If it continues to recognize self, then it can go under additional rounds of editing its light chain. And if it doesn't, then you can see how it goes into apoptosis. If it doesn't recognize self, now it can be given the cues to go out into the periphery, into circulation to the secondary lymphatic organs. And again, that's where it would co-express IgMs and IgDs. Now, another possible outcome can be that the cell will become anergic. So if the immature B cell is able to recognize self-antigen, the B cell is going to signal so that it will make IgDs instead, and that actually will signal it to become unresponsive to the antigen. It can go out into circulation, but it's not going to live long because it doesn't respond, it doesn't do anything. So anergy is where it's non-responsive, so it's still alive, but by all markers and standards. So it hasn't gone into apoptosis, but it's not able to respond to it. Okay, so again, keep in mind when these receptors are tested against self-antigen, there's 3 possible outcomes. The previous figure shows the clonal deletion where the cell goes into apoptosis, also shows receptor editing. And then the third possibility is anergic, where it just doesn't respond. So when the B cells come from the blood into secondary lymphatic tissue, you can see how it enters through the blood vessel. It's going to come through the blood vessel, which is referred to as the HEV. It is going to go out towards the cortex into the B cell area after it's gone through the T cell area. So you can see this kind of dance or loop that it makes. Then it's going to move towards the medullary cords, where it can then lead through the efferent lymphatic vesicle. So keep in mind with a lymph node, we're going to have the afferent. That's where the lymph is going to enter through. The efferent is where material exits through. So there's an entry door and an exit door. The other kind of way to get into the secondary lymphoid tissue is through blood vessels. And so the B cell is going to enter through the blood vessel and then through the HEV. Initially goes to the T cell area and then moves out to the B cell area. And this is also referred to as the primary lymphoid follicle. In that follicle, that's where it can go through additional development. So here we can see how there are cytokines that are going to attract the B cell to the secondary lymphatic tissue, but then into the follicle. When it's into the follicle, one of the important receptors that are going to be expressed is B cell activating factor receptor. So BAF is also expressed by support cells in the secondary lymphatic tissue. So when the receptor binds its ligand, that's going to signal for the B cell to mature from that immature B cell stage. Okay, so again, there's these interactions that are going to happen once the B cell has received those interactions, it's a mature B cell. It can then go back into the circulatory system and circulate in the blood, in the lymph, and then return to secondary lymphatic tissues to become activated. So it can become activated, engage with antigen in the periphery or in secondary lymphatic organs. So this slide just kind of alludes to what we'll talk about in Unit 5, where the B cell becomes activated. So you can see how when the mature B cell returns to the lymphatic organ, entering through the blood, it is going to make that movement where it goes and is in the T cell area. It can interact with the T cell area. So you can see those yellow cells interacting with the blue T cells in the T cell area, T cell zone. And then it's going to move into the B cell zone or the follicle, where it will, because it's interacted with antigen, it's going to proliferate, increase in numbers, and create these germinal centers. These germinal centers are high numbers of B cells that are proliferating. They will differentiate further into plasma cells. And you can see how we then have plasma cells that are going to go to leave the secondary organ. And so they're using the efferent vesicle as an exit. And these plasma cells are going to be secreting antibodies. So those antibodies would also be secreted out and eventually enter into the bloodstream. So if you're interested in different tumors that originate from the B cell lineage, this figure does a nice job of summarizing that. You should be aware that there are different B cell tumors, and each of them are going to correspond to a normal stage or state of the B cell during its development and differentiation. And so we can use surface markers to indicate what type of tumor you have based on those markers on the surface and what stage it is in. And then this last slide is just a nice little summary of the different stages that we have talked about. So again, think about what is going on at these different stages. Most of these stages have a early phase and a late phase. So though there are a number of stages, if you kind of think about what is the major event happening at that stage, right? So what's the major event happening at a pro-B cell stage? Well, that is the rearrangement of the heavy chain. And so the early one is early on, and the late one is, okay, we've got some of this done. And then as we move to the pre-B cell stage, we then call it large versus small. So again, what is happening at these stages? And this is where we have the expression of the pre-B cell receptor. Then we're going to start working on our light chain, and then have light chain expression. So that at the immature B cell stage, we have a B cell receptor that's going to be checked for negative selection, which isn't on here, but would be really nice if they added that, right? And then once it's past negative selection, it can go out into the periphery, the secondary lymphatic tissue, and that's where it goes through additional development at the immature B cell stage. It goes through another round of negative selection, but it's also receiving those cues from chemokines and also interacting with BAFF through its BAFF receptor so that it will become a mature but naive B cell. Once it goes out into circulation, it can interact with antigen, so it becomes an antigen-activated B cell. And then when it's returned to a secondary lymphatic organ and receives cues from T cells and additional cells, it can become a plasma cell where it can secrete antibody, right? And then even further, some of these cells can become memory cells so they can fight future infections. And again, these last three stages, so from antigen activated to memory, we'll be talking about those more in Unit 5. So focus more on thinking about how do these earlier stages align with what we were talking about with the B cell receptor. Okay, so that making of a B cell receptor and then the checking of the B cell receptor are the big pieces for this unit, for B cells. And then similarly with T cells, what are the stages of making the T cell receptor? How does that coincide with the stages that we call them? And then what are the different selection processes and location? Where is the location happening for each of these cells? All right. And as always, if you have any questions, please feel free to contact me.